And I think that's probably the explanation that requires the least amount of magic.
This is Coronavirus Part 2. I am not a virologist. I don't play one on TV. Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a bike. This is Coronavirus Part 2. We're going to get it right this time. Just as a caveat, I am not a virologist. I don't play one on TV. But I am a biologist and I can read stuff. So I have taken more time to read more stuff about the coronavirus with an attempt to try and understand what's going on with this outbreak. And try to figure out if there's any real information available on the internet that they're not giving us on TV. I think the first thing to discuss when it comes down to it is to figure out whether or not we're talking about a bioengineered virus or just a really nasty, naturally occurring one. I think this is where all the conspiracy theories spout from, and this is where all of the misinterpretation of what seemed to be nicely arranged circumstantial evidence supporting the idea that this is a bioengineered virus which escaped from this lab in Wuhan. There is, of course, the other possibility that the virus was in the lab in Wuhan, but they didn't make it. They just found it, isolated it, looked for it. And you can't get mad at the Chinese government if they were looking for novel coronaviruses in the wild, because in 2003 they had the Sars coronavirus outbreak, which infected like 7,000 people around the world, or 8,000 people around the world, and showed the vulnerability that we have and our infrastructure has to epidemic. So get out of the frickin' bike lane, you asshole. It's a bike lane. You can't just drive in it. And so determining, for example, whether or not it came from this market or not seems to be something that we should think about, because from the perspective of controlling an outbreak like this, controlling the outbreak from the source is probably the best way to do it. And so that's what the debate is about, whether or not it's an engineered virus, because the scary part would be is if they engineered the virus to be stronger to infect humans, and it's just not really very likely. And the history of these coronavirus outbreaks is what leads me to believe that that's not very likely, as well as the fact that there is a limited number of genetic tools that work on these kinds of viruses. And so that's really kind of what I want to cover on the ride, is just try to give a background on these viruses, give a little sort of biology 101 on coronaviruses, and then talk about the papers that are being used to argue that they had the technology to create a chimeric coronavirus, and then just make the argument or try to explore the argument as to why that circumstantial evidence doesn't really qualify as a smoking gun for the fact that China would have made this virus. Now, have we made a chimeric coronavirus before? Yes, we have. Do we know a lot about coronavirus? Yes, we do. What do we know about coronavirus? Well, we know a shit ton. The first coronavirus was discovered in the 60s in chickens, and then very soon after we discovered the first two human coronaviruses, which are the viruses for the common cold. These two viruses are both coronaviruses. They're both human coronaviruses. They are from the alpha coronavirus group. Human coronavirus 229E, for example, is a common cold coronavirus. The avian flus were coronaviruses. The H1N1 flu is a coronavirus. And so when I said in that first video that they took something from a Sars virus and put it on a coronavirus, I was kind of talking like an idiot, because Sars virus is simply a subname, a specific name for a particular emergent coronavirus. And what these coronaviruses all have in common in terms of what happens is that they emerge from an animal host and are able to cross over into a human. And that's what happened with SARS. And SARS binds to a receptor that is enriched in the lungs, and that's why this coronavirus and other coronaviruses oftentimes cause respiratory distress and respiratory infection, because the point of entry in the body is the lungs. And so that's where the inflammation occurs, and that's where the acute syndromes of the viral infection occur. And that's what SARS means, right? It means severe acute respiratory syndrome. Now, SARS virus was a coronavirus that emerged from the wild in 2003. Another well-known example of coronavirus emergence is the MERS, the MERS virus in the Middle East. This virus is carried by somewhere between 20 and 30% of all the camels. And so in particular risk are people who work with camels. And I think 8 times the MERS virus has emerged separately from camels to infect humans. Nasty enough, the MERS virus has about a 33 or 30% fatality rate. It's a nasty sucker, and it's still going around. But the nice thing about MERS is that you got to be close to the camels, because it has a very low R0 figure. And the R0 figure is the rough rating. Get off your phone, lady. The rough, oh my gosh, you're not even going left. The R0 rating is the rating that describes approximately how many people the typical infected person infects. And the R0 rating for the MERS virus was less than 1. So unless you lived in the same house and you had regular exposure to the contagious individual, you weren't going to get it. And that's why it's contained. That's why it hasn't spread epidemically, because it just simply isn't virulent enough. It's not contagious enough. Now, the SARS virus was more contagious than the MERS virus, but it was much less fatal. So you still had to contain it in the same way. You still had to isolate people from the source and try to stop people from infecting one another. But because the infection rate is so comparatively low, it doesn't really matter. It doesn't really matter for MERS virus. But for the 2019 corona emergence, this virus seems to be much more infectious, much more contagious and it has been given speculative R ratings as high as 6, but the ones that I can find in the literature are around 2.5 or 3. But that's still quite a bit higher. I think SARS was 1.7, so it's about double the infectious rate of SARS. Now, the point of it being from a animal is twofold, and most of it has to do with what action do you take, right? If you think it's a zoonotic event, a zoonosis is an animal disease that crosses over into humans. If you think it's a zoonosis, then the primary source of the infection is the animal. And if you think it's a human-to-human infection, then the primary source is a human. You want to win? You can't win. He wants to win, but he can't win. So that's my point. Come on, dude. Is that if they would have known that it was human-to-human transferring, they might have taken action quicker. And a lot of the publications that are coming out now are really documenting the human-to-human transfer. And that seems to be the main point, is that maybe the possibility is that the Chinese authorities didn't know and didn't expect it to be a human-to-human transfer. They thought it was zoonosis. They thought it was a zoonotic event. So as long as people weren't eating bats or whatever nonsense you think it is, as long as they weren't exposed to the wild meat or the wild products, then the virus wouldn't spread anymore. And it wasn't really until they found this family in Shanghai or somewhere that had gone to Wuhan and came back infected that they had the first real epidemiological evidence that indeed it was human-to-human transfer and that it was doing it well. And before that, given the history of these diseases, it is kind of expected that even if it crosses over into humans, it will not have mutated in such a way that it will be contagious to other humans. And it's hard for me to know exactly the difference between that, and I don't fully understand how it is that a virus can cross over into a human host but then still needs to mutate more. I'm showing you a figure now from one of Barrick's papers, and I think what the point is that if it transfers from the animal to the human, that transfer can happen and infect the human, but the mutation that's required for efficient transfer from human to human might be different than the rare mutation that just allows it to jump one time. And so if you allow it to jump one time but it can't jump again, then the virus itself needs to grow and multiply and mutate in human hosts or an intermediate host to become the extremely virulent strain that causes the epidemic. And so what Barrick makes an argument in his early paper there when he does the mutagenesis of one coronavirus to another, very specifically he takes a bat coronavirus spike protein, a novel one, and puts it on the known Sars virus backbone to show that now that spike protein allows passage into human cells. It wasn't very virulent. It wasn't a very strong thing, but it did work. And the argument was then that there are coronaviruses in bats that are a lot like SARS, and if you look at their little spike protein, the spike protein seems to be effective as a cell entry key, because when we make a chimeric virus with SARS, it works. That's what that paper is. So you could draw the conclusion that we have sufficient technology to make a chimeric virus. And so if, for example, somebody spent a lot of time isolating viruses from horseshoe bats, like they did in China, and some of the researchers that worked at the University of North Carolina also worked on isolating novel coronaviruses from bats. So this virus shares the most homology with a bat coronavirus, but where the chimeric idea comes from is that the spike protein appears to be pretty homologous to the SARS coronavirus spike protein, and not so related to the rest of the spike protein sequences that are in the small group of viruses most closely related genetically to this virus. So in this figure you see a little glob of viruses on one side and a glob of viruses on the other, and SARS is in one and the human virus is in another. And so, although it shares a lot of homology with these bat ones, the interesting part is that this spike is really similar in function, at least, to the SARS virus spike protein. And that doesn't really make a lot of genetic sense if you're trying to find the sort of genetic chain of animals and mutations that led to the current virus that we're dealing with. But just because there isn't evidence of a recent recombination event doesn't automatically mean that we built it. And there's a couple good reasons why. And the main one is that almost all of our tools for editing genomes edit DNA, deoxyribonucleic acids. And the genome of most viruses, including the simple poliovirus, all the way up to the giant coronaviruses that we're currently talking about, they all have a positive sense RNA genome inside of a protein capsule. And we don't have very many tools for editing RNA genomes. RNA is generally, in our molecular biology, RNA is often and most considered a messenger molecule. It is the bridge between DNA and protein expression. So your DNA gets transcribed to RNA, and then RNA gets transcribed into proteins, and the proteins actually do shit. So in a viral genome, the DNA is not DNA, it's RNA. And then the RNA gets translated to proteins directly, and the RNA gets replicated and packaged into new viruses that get secreted. So, because we don't have, like this thing that's in the news now, CRISPR-9, Cas3, CRISPR-9, whatever, CRISPR-Cas9. It's a highly specific DNA editing protocol that allows you to make targeted point mutations and really put in exactly the code that you want to put it in, exactly where you want to put it. That system won't help here. Cas3, according to some of my sources, could digest the mRNA, or the RNA genome, but it wouldn't assemble it in any meaningful fashion. You'd have no control over that. So to insert a particular sequence into a coronavirus genome would be very difficult. One of the reasons why, and continuing with this size thing, is that in one of these blogs we cite the fact that in 2002 we made the polio virus from scratch. That's true. But the polio virus is one of the smallest and simplest RNA viruses there is on the planet. And the size of its genome is 7,500 base pairs. And the average genome size of a coronavirus is between 20 and 35,000 base pairs. So there are more genes encoded, more proteins encoded. It's just a lot more machinery going on. Enzymes that the virus needs once it enters cells. So it's just not as simple as, making a knockout mouse nowadays. And so we shouldn't say that the tools are easy. And then the other thing is that although this new spike protein sequence resembles the SARS spike protein, there's still some differences. And those differences are not changes that a geneticist would make. And let me just justify that. We can model how proteins fold, and we can model the three-dimensional structure of the receptors to which they bind. And so we've done that with coronavirus. And with the ACE2 receptor, we've done that. And the computer models would have never picked this sequence to have a nice affinity for the ACE2 receptor, because the base changes, the mutations that are present different from the original SARS virus, are not predictable by our computer models. No gene jockey would have ever said, okay, I'm going to make this. But it works still. And so that's evidence from the structural perspective that it was probably just random mutagenesis. The viruses change all the time, we know that. That's why we get the common cold every year. That's why the flu is going around all the time. And these big RNA viruses, they mutate rapidly. So what they proposed in this 2013 or 14 Nature Medicine paper was that it could be that a lot of different variants of the virus are just floating around in these animals. And it may not always be necessary for there to be an intermediate host. It may not always be necessary for there to be further changes. Every once in a while there could just be an extremely virulent, rare virus that emerges directly from nature. The other possibility, and this is not crazy either, is that they've been actively collecting rare coronaviruses from the wild. I mean, we know that they're doing that. There's lots of papers isolating novel coronaviruses from bats. So any of these viruses that are stored or isolated in a lab, purified in a lab to study, they don't have to be genetically engineered to have stumbled upon a very nasty sucker. Oh God. A very nasty sucker in their search. And then, yes, that still says that it got out on accident. So I'm still sort of speaking a conspiracy theory. But they have had problems with protocol before. Thanks for joining me on my ride. That was my second attempt at a coronavirus video. I hope I got the basics right a lot better this time. I made some mistakes in the first video basically because I was very excited about the biology and a little overestimated how much I knew from my background. In this video I tried to give you a basic background on coronaviruses and the idea that the common cold and the flu virus and a lot of the viruses that we think about today are coronaviruses. I tried to give you an idea of some of the things that happened earlier with SARS virus and with MERS virus and why those viruses are a little bit different. SARS virus was in 2003, MERS virus is in 2012, and it's still going around, but it's not so contagious. So it's, generally speaking, although it's very deadly, around 30%, of the cases are fatal. It's just not as dangerous because it doesn't travel as fast. The Sars virus in 2013, which is also a coronavirus, was about a 1.7 R naught, and this one is estimated to be somewhere between 2 and 4. There are estimates in different papers. I've read 6, but I think the consistent data is around 2.7 to 3, which means that every person who's infected has a probability of infecting 3 other people. That's fairly virulent. The other problem, or the other thing that we talked about, and the sort of conspiracy ideas stem from this, are the idea of whether or not this is a man-made virus. And number one, I don't think that debate is really necessary, because either way, it's a nasty virus that's out there. If we want to talk about hubris and whether or not mankind is being kind to nature, that's another topic. But suffice it to say, there are a couple good arguments for why it's not a man-made virus. The first one is that the sequence prediction for the ACE2 receptor and variants thereof from other viruses known to attack it, known to target it, including the original Sars virus, the mutations that are present between that typical spike protein and the one that's found on this novel virus are not changes that any gene jockey or computer program would predict would result in equal or greater affinity to this receptor. So it's not very likely that this sequence would have been chosen by a gene jockey. So that's number one. Number two, even if this gene jockey had this sequence in mind, putting it into an RNA virus is not something that we have the molecular technology for. CRISPR-Cas9, for example, is technology that works on DNA and genomic DNA, but that's a completely different molecule with completely different helper proteins and a whole different scenario in terms of making and breaking. And here, to engineer a virus, we cited in 2002, for example, that we engineered a polio virus, but the polio virus is the smallest and simplest virus of all of the RNA viruses. It has a genome of about 7,500 base pairs, and a coronavirus typically has a genome over 25,000 base pairs. So the number of proteins that they encode and the amount of machinery that's encoded inside of a coronavirus is much bigger and greater than whatever is encoded inside of a polio virus. Couple that with the fact that the spike proteins and the outer protein code is much more complicated in a coronavirus, and then you really have an issue here where it's much more likely that nature created this virus. Now, were the Chinese mining them? Were they finding them? Were they actively seeking them for research purposes? Probably. And did they find it? Maybe. Did it get out? Maybe. But again, we're speculating about things that don't really matter. If it started in the market, then we should have been able to stop the spread much easier, because the source would have been an animal. And that's the point of the zoonosis thing, is that if the source is an animal, 99% of the time when it comes into the human, it's not contagious enough from human to go anywhere without further mutagenesis, without further changes. So it requires several sick people to get it before it can be sufficiently changed through replication and mutation to become virulent across the same species. So typically there's always like a mouse or something that crosses from the bat to the mouse to the human. And in that paper from Dr. Barrick, they make the argument that's not necessarily true, that there could just be a mess of viruses in these wild animals, and that occasionally you get a rare copy that happens to get out and it happens to be able to jump species. Because this has happened before, it happens across species. It just doesn't always go to humans. It's gone from birds to pigs. It's gone from camels to humans, and this kind of thing happens. So the idea that this happened one more time is not crazy. The question only becomes whether or not anybody was irresponsible in terms of collecting the virus, in terms of replicating the virus, or in terms of containing it. And that's always possible. That could happen in America as well as in China. No. The only question now really becomes is what's going to happen from this point on, and I stand by everything that I said in the last video. I think it's very likely, just like any other virus on the planet, you can't stop it with face masks and quarantines. It's just not going to work, especially not as far as it's gotten. So now the question is really, how far is it going to go, how many people are going to get sick, and what's the average death rate? I mean, that's the only thing we can really look at now. And as far as the the susceptibility to the virus for Asian people, I found only one reference that remotely suggests that this is possible. The ACE2 receptor is not particularly enriched in the whole body of the Asian population, but I found at least one anecdotal preprint that shows that the lungs of Asian people may be more enriched with ACE2, but that's from one man's diagnosis, so we're not really very far yet, and it's a preprint, it hasn't even been reviewed. So there's a lot more to come. I have some information in this video, or in the links too, about possible spreading and how far it's going to go, and I found some other information that I think is worth reading. I tried to summarize it on my ride. I didn't get through all of it, but the basic gist of it is this: yes, this is a disaster, but it's probably not a man-made one. It's probably just the evolution of the coronavirus, and we just happen to have a pretty nasty version right now. I wish you all good luck. Thanks for watching. Thanks for critiquing my video. Thanks for striking me on Reddit and other places to spur me to clean this up a little bit and to get the details right. I hope you learned something from this video, at least more than the last one. And if you liked it, like and subscribe, and I'll see you next time. Thanks.
These viruses enter into human cells. So that means that they can do some of the things that might be necessary for making a chimeric virus that was highly virulent. And I think that's probably the explanation that requires the least amount of magic. To really remember is that the effectiveness, and this is a good point, the effectiveness of a face mask is not to protect you from viral particles in the air. It is to protect you from taking your hands and touching your nose and your mouth. That's how most people get infected. Touching doorknobs, touching your nose. Touching tables, touching your mouth. And so if you wear a face mask, you tend to do that less. And so it's kind of assumed, I think, in terms of popular culture, that the face mask is blocking viral inhalation, but it's not. It's keeping your hands off of your face. So if you want to stay clean, wash your hands a lot, keep your face clean, stay away from crowds, don't let people cough on you, the normal stuff. Face mask is only going to keep you away from your own face. So anyway, stay safe, stay clean. I hope you got something out of this video. You should have gotten more out of this video than the last one. And if you want me to do more coronavirus videos, I'm happy to do it. Just give me suggestions in the